The ultraviolet and visible light spectrophotometer, or UV-Vis spectrophotometer, is a staple in most laboratories. It is one of the simplest instruments in terms of parts and operation, but is still composed of a variety of pieces, which will be briefly explored in this video. Many instruments in the laboratory appear from the outside to be impenetrable black boxes. In this video, the lid will be lifted to examine what pieces are used specifically in the UV-Vis spectrophotometer and how those pieces are arranged. As a spectrophotometer, the instrument is going to measure the interactions between light and a sample, so there need to be both a light source and a sample holder. To make measurements in both the ultraviolet and visible ranges, most UV-Vis instruments have two light sources: a tungsten filament lamp for visible and IR light from 320 nanometers to 2500 nanometers, and a deuterium arc lamp for ultraviolet light in the 110 nanometer range. The mirror in the light path turns to change which light source is directed to the sample, and this turn usually happens somewhere around 360 nanometers when scanning from low nanometer range to a high nanometer range. On the other side of the sample holder is a detector. This can be a photomultiplier tube, a photodiode array, or another kind of light detector. When using a photomultiplier tube or photodiode, or other detector that can't distinguish between the light of different wavelengths, a monochromator or filter is added to the setup. Both monochromators and filters narrow the polychromatic light from the light source to a narrow band of wavelengths, or ideally a single wavelength. The monochromator or filter should be placed between the light source and the sample holder, so only the monochromatic light reaches the sample. However, if you are using a detector that can distinguish between light of multiple wavelengths, such as a photodiode array, polychromatic light passes through the sample, and a diffraction grating is used to separate the wavelengths of light at the point of the detector. In a single beam spectrophotometer, the path of the light goes directly from the source to a monochromator if present, and into the sample holder. It is guided by a series of simple mirrors. Once it passes through the sample, the light reaches the detector, which ultimately measures the intensity of the light that reaches it. In this setup, instrument and solvent blanks would need to be recorded before you take a measurement of a sample. While this is easily done, it can be a concern about how the background changes if sample absorbance is being measured, such as during a kinetics experiment. Then you would need a different sample setup. With a double beam spectrophotometer, the beam of light is split and can take two different paths. There are two options for how the beam is split in a double beam spectrophotometer. In a double in space spectrophotometer, the light is split using a semi-transparent mirror where half of the beam of light will go to the sample and the other half will pass through a reference cell containing a solvent blank. The light is physically split between the two paths, which is why this is called a double in space spectrophotometer. The other option for a double beam spectrophotometer is double in time. As the name implies, this setup splits the time in which the beam to enter the sample or the time which the beam enters the reference cell. This is achieved with a rotating mirror beam splitter, as shown in the top right corner. This beam splitter is basically a spinning circle where opposite wedges are either mirrored and reflect the light toward the reference cell or open the light to pass through to the sample. In both cases of double beam spectrophotometers, changes in the solvent blank placed in the reference cell can be observed at the same time as the sample measurements are also being made. In any style of spectrophotometer, whether a single beam, double in space, or double in time, the output of the detector and the background subtraction from the reference cell is monitored by a computer. The output of the measurement can be viewed in either absorbance or transmittance, and is usually graphed as a function of the wavelength observed. Now, there are many options for light sources, monochromators or filters, sample holders, and detectors that can tailor a UV-Vis spectrophotometer for the specific needs of a project. However, the basic parts outlined in this video will be present when the cover of the instrument is lifted. Thanks for watching.